Darwinian evolution presumes that randomness is going to provide new genes for natural selection to work on. In this video, I will run experiments against the Sudoku puzzle board to see just how good randomness is at finding new genes to work on. I will say that the results of this experiment surprised me, astounded me actually. This experiment was inspired by Dr. Doug Axe's book, Undeniable, where he discusses the impossibility of randomness producing new functional genes. So let's dive in by looking at the Sudoku board. 9 cells with 9 squares each. The mission is to make each cell contain the numbers 1 through 9, and each row and column contain the numbers 1 through 9. Now the question, what if we roll a 9-sided die and place the roll in the upper left cell? Then we roll again, placing the number in the next cell. At some point we may find that the roll will break a 1 to 9 rule. If so, we start again. How many times will we need to start again before we have randomly generated a valid Sudoku board? 1,000? 10,000? 100,000? 1,000,000? More? I guessed 100,000. So make your guess. Be honest now. Remember it. Dr. Axe says that when it comes to fantastically huge numbers, we have a tendency to grossly underestimate. Well, I tested it. I wrote a program that fills Sudoku boards with random numbers and sees how many of the 27 checks add up. You know, 9 cells plus 9 horizontals plus 9 verticals. Well, I ran it 100 million times. It took about a week. How many valid Sudoku boards did it find? None. Not even close. Now, it found a valid check one way or the other, well over 2 million times. It found over 30,000 tries that produced 2 valid checks. There are 271 cases where 3 valid checks were found, and 1 example of 4 valid checks. Let's look at that one for a minute. You'll notice that the second row is valid, the second, 6th, and 8th columns are valid. But this is enormously far from being a valid Sudoku board. I don't understand. Aren't there a gazillion valid Sudoku boards? A gazillion? Yes. Over 6 sextillion. That's just less than 10 to the 22. That's way more than the number of organisms on Earth, and almost as much as all the data in all of the world's computers. Okay, so why can't we find 1? Because there's 9 to the 81 different ways to fill a Sudoku board. That's more than 10 to the 77. Well, how big is that? Well, 10 to the 69 is the number of atoms in the Milky Way galaxy. So we're talking the number of atoms in 100 million galaxies. That's a fantastically huge number. But the number we really want is the number of wrong answers per right answer. This will tell us about how many times we're going to have to guess before we even get 1 right guess. We need to divide 10 to the 77 by 10 to the 22 to find this number. So if you divide 10 to the 77 by 10 to the 22, you get 10 to the 3.5. That means we should get a valid guess once every couple of thousand. If you recall your exponential math, to divide, we subtract the exponent. So 10 to the 77 divided by 10 to the 22 is 10 to the 55. We're still talking 100,000 times more than the atoms that make up the Earth, or 100,000 times more than the number of organisms that have ever existed. So you're saying we need to run the program until we have tried 10 to the 55 tries before we can expect to find just 1 valid Sudoku board? Yes. To compare apples to apples, my computer tests about 10,000 Sudoku boards per minute, about 5 billion boards per year. There are about 2 billion computers on Earth. If we assign every computer on Earth to find 5 billion Sudoku boards per year, we would expect to find our first valid Sudoku board in 10 to the 31 years. That's 10 decillion years. Decillion? It's reasonable to say that valid Sudoku boards are not the product of random chance. Yes, definitely. But what does that have to do with biology? Well, a gene is a sequence of nucleotides in DNA. The sequence defines a sequence of amino acids that make up a protein. The order of the amino acids in the protein determines how the protein will fold, how it will become a three-dimensional object with specific chemical properties, a part, if you will. As the three-dimensional shape of a human-made part and the materials of that human-made part determine how the part behaves, so with proteins, its folded shape and properties determine how the protein interacts inside the living organism. If the protein's shape and properties allow it to play a valuable role for the organism, it is seen as functional. If not, it is unwanted and rejected by natural selection. How do the chances with Sudoku and the chances with genes compare? As a Sudoku board has 9 possible numbers in any given cell, so a protein has 20 possible amino acids at any given position on the protein chain. As there are 81 cells in a Sudoku board, so there are about 150 amino acids in an average protein. So let's do the math. There are 9 to the 81 ways to fill a Sudoku board. There are 20 to 150 ways to arrange amino acids to make the average length protein. 9 to the 81 is just over 10 to the 77. 20 to the 150 is over 10 to the 195. Okay, how big is 10 to the 195? Too big. Let's see. There are 10 to the 82 atoms in the universe, so we need the atoms from 10 to the 113 of the mythical multiverses to get to 10 to the 195. We need that many tries to try all variations of an average length protein. When we looked at Sudoku puzzles, we divided the number of possible puzzles by the number of valid puzzles. With protein, we need to divide the number of possible proteins by the number of functional proteins. Yes, exactly. But how many functional protein types can there be? Well, there seem to be less than 1 million named proteins. Many of these proteins have multiple variants. If we go with 1 million proteins and multiply by lots of variants, say 10,000, well, we're up to 10 to the 10. Are there a bunch of proteins that haven't been found yet? Probably. How big of a number can we put for unfounded proteins? Another 1 million? Each with 10,000 possible variants? So another 10 to the 10? So we have 10 to the 10 known protein variants plus 10 to the 10 undiscovered protein variants. That's 10 to the 20. No. We're adding, not multiplying. It's 2 times 10 to the 10. That's not even close to 10 to the 11. Oh, that's bad. 10 to the 195 divided by 10 to the 10 is 10 to the 185. So we'll need as many tries as there are atoms in about 10 to the 103, still over a Google mythical multiverses to get one functional protein from scratch. Ain't happening. Hey, idiot. You're like all the other idiots. You don't understand the theory. Oh, please enlighten me. New genes don't come from nowhere. They mutate from existing genes through gene duplication. Well, not exactly. Orphan genes are found every time a new genome is sequenced. Their ubiquity has been one of the biggest surprises of genomics over the last 20 years. Even orphan genes don't pop into existence from nothing. They rise out of the junk DNA. It's true that it's easy for randomness to put a start codon into non-coding DNA to see what happens. But that random start codon will invoke one chance, like choosing the start of a digital sequence to put into a Sudoku board. But it's not random. The junk DNA is a graveyard of formerly working genes. Therefore, the structure of the working gene is still mostly there. Oh, I see. If we start with a mostly correct Sudoku board, randomness should be able to find new Sudoku boards from it, right? Right. Well, let's try it. Yep, I wrote another program. This program uses one of the 10,000 valid Sudoku boards that I made and damages just one cell. Then the puzzle is submitted to randomness until it either finds a new valid Sudoku puzzle or until it fails all 27 checks. Periodically it gets back the original puzzle, but that doesn't help us. Yep, I ran it 1,000,000 times. Nothing. Bupkis. In the average of about 30 random changes, every puzzle became absolute mush. What would a unique puzzle look like? Well, where a puzzle has the right configuration, there's an average of 8 opportunities in any puzzle, then changing 2 numbers in each of 2 columns will get a new puzzle. Let's look at this puzzle. We have a 4 and an 8 here, and an 8 and a 4 here. Each of the rows, columns, and blocks involved have one of these 4s and one of these 8s. If we swap these 4s and 8s, we get a unique puzzle. That didn't seem hard. Randomness can never find one of these new Sudoku puzzles? Not quite never. My calculations say that an alternative puzzle will be found once in just over 1 billion tries. And this is similar to what Dr. Axe reports in his book, except you don't normally get new genes from just 4 changes. He and Dr. Gager carefully tried to convert the KBL gene to the BioF gene. This apparently happened in nature via the duplication and mutation model. Their conclusion was that it wouldn't happen by chance more often than once in 10 to the 72 tries, i.e., not by chance. So as I see it, not only is de novo genes by chance falsified, but duplication and mutation by chance won't produce new genes either, only new variants of the same gene. In truth, I view the exalted role given by Darwinism to randomness as nothing less than magical thinking. But Bruce, yes? How did you get the 10,000 valid Sudoku boards that you were experimenting on? I wrote a program. How does it work? I put the numbers in the cells one by one. Each time I check to make sure that the number won't break Sudoku. If it does, I try a different number. At some point, I usually find that no number will work. Only then do I start again. About once every 1,000 tries, I find a valid Sudoku board. So you make a change, then you see if it's valid. If it is, you keep it. If it isn't, you try again without throwing away your previous work. Yes. Isn't that how Dawkins' Weasel program works? Close enough. So you just answered your own question. You just showed how to use natural selection to find valid Sudoku puzzles. But natural selection doesn't work that way. Natural selection can only decide whether the completed package is functional or not. Are you saying Dawkins is wrong? Yes. Ah!